The B cells are the major players of the adaptive immunity because they produce antibodies. However, the B cell does not directly produce antibodies. So let's look at how antibodies are actually produced in the body. So this is a structure of two cells. Let's assume this is our B cell, right? And this is another type of cell. We call this the T cell because the B cell cannot be activated without the help of other cells. So the T cells help the B cells to be activated. So let's look at how this happens. This is the B cell. So let's label this the B cell. The B cell. And this is our T cell, right? T cell. Okay. On the surface of the B cell, there are characteristic molecules. There are several molecules on the surface of the B cells and also molecules on the surface of the T cells that enable the activation of B cell. The first of them is this one I have drawn, and these are called surface immunoglobulin. They are called surface antibodies, or you can call it immunoglobulin. Surface immunoglobulin. They line the surface of the B cell. Now, if you look at the structure of the immunoglobulin, I've done a video on the structure of immunoglobulin. You can go and check that out. We have the variable region and the constant region. So the part below is the constant region and the part above is the variable region. And I mentioned that it is called the variable region because it has the ability to class switch. It has the ability to change its form to accommodate any type of antigen that comes into the body. All right, having said that, the next molecule that we can find on the B cell is what we call the major histocompatibility complex. So let's assume this is the major histocompatibility complex. So MHC class 2. Class 2. The next molecule you can find on the surface of the B cell that is associated with its activation is what we call the CD40 ligand. So let's draw that up here. So let's assume this is the CD40 ligand. So which other molecule can you find here? Well, as we go in course of the video, we will look at other molecules that will be synthesized as a result of the activity of the B cell, of the T cells rather. So on the T cell, we have a very important molecule on the surface of the T cell. It is very important. It is called the T cell receptor. So we call this our TCR. So our T cell receptor on the surface of the T cell. Then we will also have a complementary molecule on the surface of the T cell as well that will complement CD40 ligand. And that is what we call the CD40. So we have the CD40 on the surface of the T cell, the CD40 ligand on the surface of the B cell. All right. Another type of molecule we will have on the surface of the T cell is the characteristic molecule. And that is what we call the CD4. And this is what makes this T cell a T helper cell. Remember, we have two populations of T cells, the T helper cell and the T cytotoxic cell. So whenever you see CD4, that simply makes this cell a T helper cell. A T helper cell. So this cell is a T helper cell because it has CD4 on the surface. So if it has CD8, what will it be? It will be the T cytotoxic cell. I've done a video on the development of T cell. You can go and check that out. So now let's look at the mechanism of the whole process of activation. For the B cells to be activated, right, there must be an interaction between an antigen and the B cell. So let's assume that this surface immunoglobulin that acts as a receptor binds to a specific antigen. Let's assume we have a bacteria, right, binding to the surface immunoglobulin. The B cell has the ability to internalize it. So what happens is that this bacteria is internalized. So it is brought into the B cell. And when it enters the B cell, what happens is that the B cell begins to shred this organism into smaller parts. Okay. Now, after the shredding, it has the ability to pick one of the fragments, one part of the fragment, and load it on the surface of the MHC. Now, that brings us to another thing. The B cell is categorized as what we call APC, which is antigen presenting cells. So we have a couple of antigen presenting cells. We have the B cell is part of them. We also have macrophages as an APC. Macrophages. We also have dendritic cells. So all these are categorized as antigen presenting cells. All right. Now, the B cell presents this antigen, loads this antigen on the surface of the MHC class 2 molecule. Now, I always tell my students that T cells are blind cells. They don't really recognize antigens, except they are being presented. Okay, so T cells cannot outrightly locate an antigen and kill the antigen. It doesn't recognize the antigen, except the antigen is presented on the surface of an MHC. So that is what happens here. So when this antigen is presented on the surface of the MHC, the T cell gets to work. So the first binding that occur, the first and most important binding that occur is between the MHC class 2 and the TCR. Now this is the most important binding here. However, we also have some complementary associated binding, like the binding between the CD40 on the surface of the T cell and the CD40 ligand on the surface of the B cell. Okay, so when this binding occur, this binding also occur as a complementary binding. But what is the function of the CD4? The CD4 is very important because it checkmates the binding between MHC class 2 and TCR. The moment the CD4 notices that the binding between MHC and TCR is strong enough, okay, the moment it notices that it is strong enough, the T cell begins to secrete cytokines, begins to secrete some chemokines. And the chemokine it secretes is what we call interleukin 4. So it secretes interleukin 4. Now the interleukin 4, which are chemokines, goes to the B cell and fixes itself to the B cell, to a receptor on the B cell. That receptor is called the interleukin receptor. Okay, so the T cell releases interleukin 4. It goes and bind to interleukin receptor on the B cell. So when this binding occur, every other binding is good to go, right? So we have the CD40 ligand binding with the CD440. MHC, TCR, then CD4, causing a strong binding to happen here, right? And releasing cytokines, specifically interleukin 4, that binds to the interleukin receptor. Now, when that happens, that will lead us to the particular type of cell that actually produces the antibody. Remember, we said that the antibodies are not produced directly by the B cells. The B cell has to undergo some sort of differentiation to a particular cell that helps it produce antibody. So, when this interleukin binds to the interleukin receptor, it begins to shuffle the genetic material, it begins to shuffle a lot of things, activity begins to happen inside the cell. Okay, so we have different things happening to produce different type of antibody. Now, this is where it gets tricky. An antibody, an antibody mediated defense is a specific defense. That means that this B cell will only produce antibodies specific to this particular bacteria. It cannot produce any antibody that will kill any other type of bacteria. So the antibody that will be produced as a result of the activation of this B cell will be specific to this particular bacteria. That's what we call antibody specificity. So when the different arrangements happens in the cell to produce the different classes of antibody, because we know we have different kinds of antibody, G, A, M, E, D. So these are the different kinds of antibody: antibody G, antibody A, antibody M, antibody E, antibody D. If we are looking at antibody M, right, there are different other subpopulations of antibody M. It is not just one type of antibody M we have. Okay, we have different subpopulations of antibody M. So this inside the cell, we will have the different subpopulations of antibody M being produced. Okay, so when that happens, this B cell will differentiate into two types of cell. The first one is this one, which we call the memory cell, and another one, which is actually what produces the antibody. And this is called the effector cells. Effector cells. If you don't call it effector cells, you call it the plasma cell. So the antibody is actually produced by plasma cell. Now the plasma cell begins to secrete these antibodies, specific to that particular antigen. So they secrete it in large number. Now the antibody in turn enters into the circulation looking for this particular bacteria. The moment it gets it, it marks it for destruction, either by opsonization or by any other means. So this is an overview of how the B cell is activated.